application for stable isotope analysis in fish eye lenses, as I think everyone in this room has probably been introduced to at some point. So for fisheries stock assessments, we need a lot of information about the fish species and the populations. One of those pieces of information that we need is where these fish are spawning, how many of them there are, and even in a species as conspicuous as Goliath grouper, we only know a handful of spawning sites, and there are probably more distributed throughout our coast. And so this is a problem for a variety of important fishery species. Even if we do know quite a bit about the spawning locations and populations of a particular species, we don't know that much about how their larvae get from where they're spawned to where they start to grow up. So in this example here, we have gag, which are spawned somewhere out in this area. We also know that their juveniles spend at least a year in the lower estuaries all along our coasts. We have very little information about how they get from one place to the other. There has been one modeling study to suggest that they're riding bottom currents, but that's about all we know at this point. In order to get sort of information directly, we need to do ichthyoplankton surveys, which can be pretty labor intensive and expensive. Basically, the process is you go out on a ship, you tow an extremely fine mesh net through the water, then you look through that sample and try to find all the larval fish in there, and then try to identify them, which, by the way, a lot of the species look really, really similar as larvae as well. So I'm investigating the use of a historical perspective to look at, to use the eye lenses from juvenile fish who have already settled into their juvenile habitat and look backwards in time at where they spent their time as larvae or where they were spawned. So from Amy Wallace's work, we know that fish eye lenses do record the stable isotopes from their environment over their entire lifespan, both carbon-13 and nitrogen-15. And as they grow, they add material to that eye lens. So the smallest inner part is from the earliest time in life. The outer part is from the most recent time in life. We also know from Jen Graneman's work at about the time scale that this material is laid down. From a captive study, she determined that about 30 days, about a month after a diet switch, the new isotopic signature was 100% incorporated into that eye lens. So for my work, what I did was collected juveniles from 4 different reef fish species in their juvenile habitats. The 4 species I have here, black sea bass, red grouper, red snapper, and gag grouper, all collected as juveniles somewhere between 1 and 2 years of age in their juvenile habitats. I then peeled their eye lenses to all the way back, and I'm interested in the smallest little marble that's about 0.5 millimeter in diameter. Ran those for stable isotopes using the isotope ratio mass spectrometer here, and got information about both carbon-13 and nitrogen-15 within those eye lenses. Now, as a backdrop to this work, Kara Radaba developed an isoscape for the West Florida shelf during her dissertation, and the general trends are very robust for a variety of fish species, with carbon trending from low to high from offshore to inshore, and nitrogen trending with higher values of nitrogen-15 in the northern region, lower values in the southern region. So my next several slides will have graphics that look something like this. And what I have is an isotopic biplot with nitrogen-15 on the Y, carbon-13 on the X. And since on the West Florida shelf, these generally apply to higher values of nitrogen to the north, lower values to the south, I have the secondary axes generally describing that trend. Here's the data for all 4 species. Generally, they use pretty similar habitats. They're all basically living on the West Florida shelf. Okay, great. So the next thing that I did to parse this data was use a statistical package created for R called SIBER, which stands for stable isotope Bayesian ellipses in R. Essentially what it does is create a standard ellipse around the data encompassing 40% of the central 40% of those data points. There are reasons for that. I can discuss that later. But what we find is that you can get not only a location for that standard ellipse, but also a size. And the size in my analysis here is basically corresponding to a general geographic area. So here's black sea bass. In contrast, here is a standard ellipse for red grouper, about half the size, and shifted substantially to the south and offshore from what we see in the black sea bass data. Red snapper is much more elongate in a north-south direction and very constrained in an east-west direction. And then finally, gag are similar in size to red grouper, but shifted slightly to the north. A more simplistic analysis of just mean and standard error for each one of these species sort of corroborates these general trends as well. You have black sea bass furthest to the north and inshore, red grouper furthest to the south and offshore. Now, the interesting thing about this is sort of in a general way, we can start to place these species spawning locations onto a map of the region. I showed you before, we have a pretty good idea of where gag spawn, and so we can put the central area of gag spawning right in that area, and then place the other three species in relation. We also know something about the other three species and where they're spending their time as adults, generally where they're spawning. And in all three other cases, this does corroborate some of what we know from the field. The central region for black sea bass is in that armpit. Thank you. Like, what's the real name for that place? The Big Bend region. The armpit. The Big Bend region. We have recent data that suggests red snapper spawning right here on the edge of the West Florida shelf, slightly to the north of where we know gag are, and also red grouper as adults are generally found south of all three of the other species. So this does seem to really corroborate a lot of what we understand from the field. Another interesting thing we can do with this SIBER analysis is look at the overlap between the standard ellipses for pairs of species. So in this example, we have black sea bass and red snapper with almost a 50% overlap, suggesting that their larvae are using broadly similar areas of the West Florida shelf, whereas black sea bass and red grouper have a very tiny amount of overlap, suggesting that these guys are very separated in their use of the West Florida shelf. The next thing that I wanted to look at was within species variability with larval origin for these juvenile populations that are spread throughout the region. So the two data sets that I have here to compare are the juvenile locations. So these are the locations where the juvenile fish were captured, and the isotopic values from the inner islands core representing that larval period. And so what I did was a Pearson correlation between the latitude and longitude and those isotopic latitude and longitude of captures for the juveniles, and the isotopic values, both the nitrogen and carbon, for those nucleuses. So in my example here of red snapper, we have highly significant positive correlations between nitrogen and catch location in both latitude and longitude, significant negative correlations between carbon and the catch locations, suggesting that possibly there are multiple different spawning locations for red snapper distributed throughout this region and seeding a variety of different juvenile habitats. In contrast, red grouper has no correlation between isotopes and location and juvenile location, suggesting that there's potentially one core area for spawning, and juvenile habitats are all seeded from that single area. So in summary, the island's nucleus isotope values do differ by species, both in extent and their location isotopically. And these results do seem to be consistent with previous descriptions of all the species that I used. There is evidence for potentially multiple different spawning sites seeding different juvenile locations in some species, but not in others. No scientific work is ever finished. There's always more that can be done. And of course, refining analytical precision is number one priority, both in mechanical peeling of the lenses themselves and because we have a new IRMS that actually has can use a much smaller sample. We can get an even finer resolution to this data. I'd also like to cross-reference the information that I have with egg and ichthyoplankton surveys to better understand the actual geography of the situation, and then expand to additional species. These are pretty good species that we know a lot of other things about, and expanding into species that we don't know so much about that we could be testing this with is a definite direction to move with this work. Thank you.